All right, let's get started here. I'm Dusty Majumdar, CEO of PredxBio, and we have a wonderful session coming up here. As you can see, we have a very distinguished set of panelists that we will get into in a few minutes. All of them are here. Laura Dillon from AstraZeneca, Dr. Arutha Kulasenge from University of Queensland, Mike Sorachi, Tulia Bruno, and Chakra, the founder of PredxBio. The theme for today, can you hear me okay at the back? Okay, just let me know if I have to yell more. The theme, this microphone is, the theme for today is harnessing spatial multiomic biomarkers and ushering in a new era of spatial medicine. We're going to start off with a little bit of introduction to what we do at PredxBio, and then we're going to have Chakra come and talk a little bit more about the technology, and then Arutha Kulasenge, Dr. Arutha Kulasenge from University of Queensland, is going to talk about some recent results that we have. Some really amazing results exposing the mechanistic underpinnings of cancer and how tumors interact with immunotherapy. And finally, we're going to have a panel. We're going to try to have the panel for 30 minutes, the last 30 minutes of the hour here. So really excited about it. And if at any time you cannot hear any of us, just yell from the back, because I'm hearing a lot of buzz, so I don't really know whether I'm being heard or not. All right, so we also have two posters coming up, one tomorrow and the one day after. Again, some very exciting work that we have done with Nikon, Lunaphore, as well as with Dr. Arutha Kulasenge. So very exciting stuff that you can all attend in the next 24 to 48 hours. So at PredxBio, we've been around for almost 6 years now. And what we do is come up with next generation spatial biomarkers. And Chakra Chandabotla started in this path around 10 years ago, and very proud to see that now we're getting recognition in journals like Nature, which are carrying full issues on spatial biology. Spatial biology was not known, as you all know, around 5-6 years ago, really. No one talked about it. And today, I was counting, I at least counted 35 different booths which had messaging around spatial biology. So incredible advance over the last 5 years. You know, one of the things that's it's a bit incredible, but we take pride in the fact that spatial and biology, these two terms were brought together, if you do a PubMed search, in a publication that Chakra had in Nature Medicine in 2020. That has spawned around 2,500 papers since then. Nature Methods recently declared spatial biology to be the method of the year. And PredxBio, if you look at the top, we have published around 25 different publications in marquee journals over the last few years. So the problem that got us thinking in this direction was that with the genomic expression biomarkers, PD-L1, et cetera, the response rate to immunotherapy for patients was in the sub 20% range for the last decade or so when we looked at it. And this really has not changed even now. This got us thinking on how to really identify the right patients at the beginning for immunotherapy. And we also realized that the failures that we saw in the clinical trials among the pharma companies, 90% failure. We can debate about the number, but it's probably between 80 and 90% in terms of cancer trials failing even now. Those are related to the fact that there is no real good biomarker to identify patients who would respond versus patients who would not respond. And this is what we do. We get away from the historical approach, and we use our spatial biomarkers to separate out responders from non-responders, the diagram on the right-hand side. Once we do that, we get better stratification and selection of patients. We get drugs that meet their endpoints. We have worked with a number of pharma companies to demonstrate that. And ultimately, we get great companion diagnostic tests as well. So that's basically what we do. The difference is that we've been using spatial biomarkers now for the last 5 years. We've also shifted the focus to phase 1 in clinical trials. And we've had a number of discussions with our partners in AstraZeneca, like Laura and Mike Sorachi, Laura Dillon and Mike Sorachi, that really we should try to expose the mechanism of action in phase 1. And then, you know, if it's a complex biomarker panel in phase 1, deplex it to get something simple in phase 3 so that it can be deployed, it can be robust, and it can be cheap. So we'll talk about some of the deplexing efforts that we have had over the last few years. And initially, we looked at immuno-oncology mainly. Now we have spread it to ADCs, multispecifics, and these are, as you know, the emerging category of therapeutics. The results that we have got now for across a lot of solid cancers, as well as looking at lymphoma, when you look at the bone marrow, the same patterns emerge with spatial biology. And what we have seen is around 90% accuracy in terms of predicting response to immunotherapy and other therapies versus what is available out there, the PD-L1 tests that have been around for 12 years, and the only approved test really out there for immunotherapy at this point of time. So this is what we have been doing, working with pharma and some of the research groups, powering precision therapeutics through revealing cancer biology with actionable AI-driven spatial biomarkers. So I think 10 years ago, Chakra had the realization that heterogeneity in the tumor microenvironment is not random. There is an organizing principle to the heterogeneity that we see. And Chakra realized around 10 years ago that the microdomains, which are spatial constellations of cells that come together in a specific orientation or configuration, are the key to defining if the tumor is going to respond to therapy or not. Once we're able to identify these microdomains, we can actually get to the network biology of cancer, and then it also holds the promise down the line of in silico clinical trials. So, as I said, these microdomains are clusters of cells coming together. They're repeatable, and they are distinct cellular, spatial, and functional patterns which drive cancer progression. Once we identify these microdomains with our computer vision and AI software, we are able to predict if the patient is going to respond to therapy or not. And from the microdomains, we go to explaining the network biology of cancer. Now, this has been around for a while. A lot of people talk about all these different kinds of cellular neighborhoods and cellular niches. Robert Weinberg in 2018 in a paper actually pointed out that spatial information paired with immunological composition and cellular status can help identify the presence of micro niches. But the interesting thing is, nobody really has been able to quantify this in the way that we have and also correlate it to response with around 90% accuracy. So that's what we're here to talk about, show you some examples of what we have done. Most of you are familiar with the spatial imaging platforms. These actually help to tag the biomarkers with different markers, the antibodies. And then we start our magic in the SpaceIQ PredxBio platform. We're able to capture protein interactions, the cell-cell communications, as well as these microdomains, which are clusters of cells coming together in a specific spatial configuration, which are identical. And once we identify the predominant group of microdomains, we can predict response to therapy. So one of the things that we have done right from the beginning with Chakra is we've integrated the AI and cancer biology. So we don't use just a black box for deep learning or machine learning that 99.99% of different companies do. We try to integrate the biology. We do all the exploration, for example, discovering the microdomains in an unbiased way without imposing a hypothesis. And ultimately, we are able to get to the network biology of cancer. You see lots of examples of that. Now, there's been a plethora of papers recently, even talking about futuristic stuff like the AI virtual cell and foundation models. And there are some folks here talking about spatial agents as well. All of those have their place. But if you're looking to have a fundamental evolutionary block of cancer that's going to predict how phase 3 trials are going to work based on phase 1 data, unlocking the reverse translation and revealing mechanisms of action, identifying novel targets, we're able to do that at this point of time. Here are the publications, heavily patented our technology over the years, and thanks to AstraZeneca, a wonderful figure here, where with a multiplex spatial biomarker, which could be 20 or 40 plex, we're able to reveal the mechanism of action here. That's one of what we do in phase 1, 2. And then we go to phase 3, where we actually can deplex the complex biomarker into low-plex spatial panel, and which is going to be robust and deployable, cheap. So let's take a look at our PredxBio platform. We're able to ingest data from pathology, spatial proteomics, spatial transcriptomics. Once we do that, we are able to phenotype after some basic quality control, as you can see here, automatically and in an unbiased way, do the functional cell phenotyping. Once we get these different phenotypes, we are able to use our computer vision software and find these microdomains that we talked about. And these could be a combination of tumor cells, fusion cells, macrophages, et cetera. And once we find the microdomains, as you can see on the right-hand side, we are able to go to the next step and get to the network biology of cancer. So that's kind of what we do, and we try to address some of the most challenging problems that are out there. For example, predicting with 90% accuracy from phase 1 to data, phase 3 trials, exposing the mechanism of action, integrating spatial multiomics, proteomics, and transcriptomics data, and optimizing complex biomarker panels in phase 1-2 into much more simplified panels. Here are some use cases. Chakra and Aruta will talk a lot more about this. This is really an astounding use case that we're very grateful to Dr. Aruta Kulasinghe for working on, where we're able to identify responders versus non-responders based on spatial combination of metabolic biomarkers. We started off with a 44-plex MIF panel. As you can see, some of these are immune lineage, some of these are structural, some of these are metabolic, some of these are about immune activity. And the first thing, if you remember, what we do is recursively identify the full spectrum of cellular heterogeneity with unbiased recursive phenotyping. And we keep on dividing the cells till we're able to assign distinct phenotypes. Once we assign the distinct phenotypes, we're able to get the microdomains and the microdomain response family, for example, on the left-hand side, and the resistance family. Once you get the family of these different microdomains, we try to search for which of these will predict most effectively the outcome, and then we identify the one that's going to be, for example, on the right-hand side, the most effective one for resistance. And once we find that, we're able to predict with AUC values of 0.90 the response to therapy. And also on the bottom right, the network biology of cancer, differences between responders and non-responders. You'll hear a lot more about this from Aruta, so I'm going to wrap this up here so that Aruta has enough time to talk about his findings. This is another example for diffuse large B-cell lymphoma. Again, 93% accuracy here. We have done the same thing for colorectal cancer, as you can see here, with around 93% accuracy predicting CRC progression, again from those microdomains that are discovered from the spatial analysis. An interesting study here where we were able to deplex, starting with a 40 to 60-plex MIF panel, all the way to a combination of 2-plex IHC and H&E. As you can see, we've maintained very well the AUC values, starting with 0.91 all the way to 0.83, so not a whole lot of decrease in the AUC values. And that is because we understand the MOA when we start off. This is a very recent study. Aruta will talk about the head and neck cancer, and again, some great results that we have. You can see the AUC values of 0.93 and unearthing of the network biology of cancer. So just a quick reflection before I hand over to Chakra to come and talk about the technology here. 30 years ago when I started in this field, disease was defined by cancer was defined by the tissue of origin. Sam Hanash, who's here from MD Anderson, probably remembers from 30, 40 years ago, it's lung cancer or pancreatic cancer or whatever, right? Then came genomics, and we were defining diseases by mutation. And that was great, but that really, was enough to help maybe 20% of patients. And now we are going to a point where we are defining the disease by spatial multiomic signatures. And the hope is that that's going to cover a lot more patients who have cancer, and we'll be able to get to some solutions for them in the very near future. So with that, I will invite Chakra to come up and talk about the technology a little bit more. Here is kind of our booth. You can come and take a look at the astounding spatial microenvironment on the Apple Vision Pro. Please stop by at 3252. Chakra, go ahead.
I'm going to have to match Dusty's energy. I think just a quick thought. So 2 things I want to focus on. Microdomains. There's a lot of question on explainability. Where is that coming from? And as Dusty pointed out, we have a booth. We have a deep dive that we can take if you stop by. So just a few, big picture items is all I'm going to cover. And when we designed this platform, the SpaceIQ, there were a couple of things that were very much in our mind, which is that the platform should be able to help you verify the hypothesis that you have, and also be able to generate new hypotheses, because there's a lot of emergent biology that you care for. And then we wanted the software to have a broad platform coverage. And that was actually a very useful exercise. So we think that we know a lot more about every platform out there than when we started. And then you want to be very good with how you think about these signals. You have to build trust in this data set, and you want this to be reproducible. So you think a lot about that. And then there is going to be uncertainty. So you have to find a way to handle that uncertainty from the time you pick up those photons and then take them all the way to the pathways, right? So, and we're all been in this space wherein you spend a lot of time trying to collect good data, and in the end, if you're, for whatever the constraint, you're not spending as much time in actually interpreting it. So there's a huge imbalance that we want to correct with the SpaceIQ platform. So you spend actually more time interpreting the data, and then we have the tools to enable how you can reduce all the effort that goes in to clean up your data sets, right? So two pieces, hypothesis generation piece and the hypothesis verification piece. Lots of tools, modular, scalable, lots of APIs. Again, come to the booth. I'll take you through a whole lot of them. Just to highlight things like how you clean up the QC, QA, SpaceIQ platform has a way to mitigate all that. So here's some of the processing that gets done with these images in an unsupervised manner. And then off-the-shelf cell segmentation, I can't find one person who is happy with the segmentation that they work with. So here's a case wherein if the tissue is dense, clearly you lose out on a lot. So you have to bring in the biology. You have to be aware. The algorithms have to be aware, in other words, the fact that this tissue is in 3D, but the image is in 2D. So how does that really affect and impose constraints in how you do the segmentation? So SpaceIQ thinks a lot about the platform, lots of modules to think about this, and do a damn good job with the segmentation. Come to the booth. Let us take you through some deep dive. And I want to now focus just on two things. How do these microdomains come about, and what can you do with them? So this is the case that Dusty already mentioned. And the idea with the cell typing, and again, there are dozens of algorithms out there, but what we guarantee with this platform is that the decomposition is numerically stable. And just a very quick way of seeing that is every node has a color. If it is a green color, that actually I can point out that that's a very stable cluster, and there's no need to rethink about what's going on there. And the second thing is that it comes out, the biology speaks for itself. So this is a case wherein we know what biomarkers are being utilized to make what segmentation and what phenotyping possible. In this case, the metabolic markers, the SDHA and ASCT2, make a difference in highlighting the cells on the right side. This is just a painting of how much heterogeneity is there. And then the first tempting thing to do is just to count the number of them and see whether that's good enough to be able to tell the responders versus non-responders. Very often, that's never good enough. And then you're really asking, you have to go back and think about what the platform can offer you in terms of communication and quantifying cell-to-cell communication. Here, we have a very unique perspective. We go back to the very basics. Regardless of the machine learning algorithm in place, the question that you're asking is that if there is a tumor cell, can this tumor cell anticipate the presence of a macrophage? So this is about predicting the identity of the nearby cells. It's just not co-occurrence. It's really about prediction. Huge difference, huge nuance. Come talk to us as to what that means, because then you have this way of thinking about what interactions really show up as being differential between the two cohorts. So the red lines and the blue lines are a way of communicating what is different between the patients that recur and for the patients that do not recur. And what happens is that you can take any one of those. Every one of those lines is a prognostic model. So here's a case where we connected, there's a microdomain with 3 of these cell types. And you can actually sit down, interpret it, because that explanation is coming through as to how these cell types have been defined. So here's a case wherein the tumor cells, which are the very dark, bright green, they're surrounded by this fibrotic wall and also the macrophages. And you'd like to think that that's effectively creating an immunosuppressive environment to do so, right? And then there's a huge difference. One is about finding which cell is next to one another, to the other cell. But the picture on the right says, what is the reason? What is the mechanistic reason why these cells, and what exactly is the communication? So some very interesting ways to think about novel drug targets and such. And then what happens with every one of those lines, as I said, is a prognostic model. So you can build a family of prognostic models from which you can pick and choose the sort of performance that you think is important for your data set. You can map it to the pathways, as I said, for novel targets and repurposing. And then finally, the same idea of microdomains also helps you put together or integrate this multi-scale data sets. In this case, there's a single cell and spot level transcriptomics data, and there's a spatial proteomics data, and the bright field pathology data, and they're not necessarily consecutive sections. So how do you make the HND speak to all these data sets and relate those properties in HND and identify some new biology? We can do all of that. Again, come talk to us. We have 2 deep dive dates, 28th and 29th. Thanks, Dusty.
Thank you, Chapra. So we are delighted to have Dr. Arutha Kulasenge from the University of Queensland, our key collaborator for the last few years. And Dr. Kulasenge is probably the #1 KOL today in spatial biology, at least that's what I think, and many others think. So welcome, Arutha. Delighted to have you, please.
Thank you, Dusty, for the invitation. I live in Brisbane, Australia, and so making it to AACR every year is quite a challenge. And so we'll talk to you about some of the really exciting work we've done working with Chakra and the team over the last couple of years. So I'm based at the Fraser Institute, University of Queensland, really exciting sort of lab, leaning on the clinical end and translational end, where we're trying to understand predictive biomarkers for checkpoint inhibitor therapies in lung, head and neck, and a number of other tumor types. So Eric Topol, I wish we coined this earlier, and he coined this spatial medicine term late last year, and I think this is where the field's really heading. This beautiful shoreline where he explained the skin cancer story really well, and so nature took this and then they leaned on it, and they're calling it tumor geography. And so spatial is nothing new to us, but spatial medicine is potentially where this will hopefully go in the near future. This audience doesn't need to be told why checkpoint inhibitor therapy is important, but it works in a number of tumor types where a subset of patients respond to checkpoint inhibitor therapy, and really defining these is really important. An article a couple years ago showed that I did my PhD in the liquid biopsy space, but realized really early on understanding the tumor and the tissue, and then that's fundamentally where immune cells act, is really important. So getting that visual data simultaneously with your molecular information is really, really important. There are a number of predictive biomarkers in this space, PD-L1, TMB. They're limited in their dynamic range and how predictive they are. And what we try to do in the lab is to mine every cell within a patient's biopsy, surgical resection, and so on. And so why this is important is if we looked at it using a bulk expression analysis, we'd get very similar scores, but you could see on the right the tumor is highly infiltrated compared to the one on the left. So getting the visual information simultaneously with your molecular information is important in predicting these clinical endpoints. These are some of the studies that are ongoing in the lab in lung cancer, head and neck, both retrospective and prospective studies, in breast cancer, colorectal cancer, and we're looking at multi-site, multi-institutional studies where we're trying to develop robust signatures. It's not about N of 1 studies, N of 2 studies, which you'll hear a lot about. It's really about how does this scale across institutions and so on, and we do a lot of work in infectious disease as well, looking at the impact of viruses across multiple organs. And so working with Chakra and the team, one of the challenges we found was in scaling spatial biology, the informatics companies out there they are very black boxy, and so how do we sort of get a lens a bit deeper and try to understand it? And one of the delightful things about working with PredxBio was we were really able to get a feel for how this is done, and it was an ongoing discussion across multiple data sets. So this is in a lung cancer second-line IO versus standard of care cohort where Dusty spoke about the recursive cell typing. So we had responders, non-responders to second-line IO therapy, and we could start to contrast. I love the visual data, right? So I love numbers too, but actually seeing it on tissue is fundamentally where we can start to interact with pathologists and really understand what we're looking at. And so this is in resistance. This is just two examples. In resistance and response to therapy, you can clearly see the microdomains that are identified in these tissues are completely different. And we can see this across the entire cohort. So these are tissue arrays where we're doing about 45 patients per slide, single biopsy cores. And these are pre-IO specimens where we have the PFS data and OS data for all patients. And so we can start to contrast the microdomains between the responders and the non-responders. And the panel that we used was not just based on immune cell function, it wasn't just immune cell status, it was looking at both tumor markers, immune markers, functional status, metabolic activity. And so we tried to use this sort of multiomic level of information to be able to subtype, because we think at a phenotype level alone, nothing's going to pop up. You really need to dig into the subsets of the cells, and the only way you can do that is by functionally annotating those cell types. And so we can start to see that there are these clear distinctions between your responder and your non-responder microdomains in these tissues. And for example, we're looking at here in the tumor, ECAD positive, pancytokeratin positive in the tumor, but also the HLA status, proliferation, and SMA. And what we find is that in the patients that are resistant to checkpoint inhibitor therapy in the second line setting, we find that these tumor cells have these fibrotic walls on the periphery that are preventing your immune cells from infiltrating. And so you would have heard from Joanna Joyce's talk this morning, beautifully showing that, scarring and, this fibrotic activity. And it's not just cancer. We see this in the lungs of COVID patients, right? There is this fibrosis at play that's defining resistance to therapy, but we're also seeing it in multiple other conditions. There's actually an association of having fibrosis and your long-term sort of outcomes to therapy, irrespective of disease. And so we see these fibrosis-like signatures that are defining resistance to checkpoint, and then we can contrast it to patients that are actually responsive. And so the microdomains that pop up here that are in this responsive phenotype is a citrate synthase positivity in your immune cell subsets. And so it's actually showing that in patients that are responsive to therapy, we're seeing higher metabolic activity, specifically in the immune cells. And so when we contrast this across the two, you see on the left there, those are patients that are responsive to therapy versus those are patients that are resistant to therapy. But if you think about this, the markers that we're using to define both response and resistance are not 40, 50, 60 colors. They're probably 3 or 4, right? So how do we define a minimal signature for response and resistance? And I think there's some lessons in here, and we're demonstrating proof of concept, but obviously we need to scale some of that information. And so we can start to look at this at the cohort level in responders and non-responders. TMAs are great where you can get a lot of tissue on a slide, and so we can identify the microdomains here between the groups. And then these were patients that were given Nivo, and so we can start to look at the mechanistic properties of them, and on these circus plots, how they associate with both response and resistance to therapy. And so Chakra spoke about this earlier, and Dusty. So we find in this cohort of patients that there's this response feature that we're identifying in this metabolically active subgroup, which are present in an immune cell type, but then also this resistant population which is associated with this fibrosis, and we think it's linked to an EMT-like transition. I'll quickly go into the head and neck just to end. And so head and neck's on the rise. It's sort of opposite to cervical cancer, where we're seeing a lot of sort of early stage patients with oral lesions, 20, 30-year-olds with oral lesions, and we're really trying to understand what's going on in these tumors. This is in a cohort of patients that have received immune checkpoint inhibitor therapy, and these patients had recurrence or didn't have recurrence, and so we were trying to contrast the two. This is now looking at the recursive cell typing. I won't go into this, but what we wanted to do was, visually look at the cause. This is again tissue arrays between patients that had recurrence versus patients that didn't have recurrence. It's almost night and day when you start to look at these tissues directly and really appreciating the microdomains that you can now map back onto the tissue. And so I asked a really simple question of chakra. When we go into IO studies, pathologists will say, where are the CD8s? And so when we look at the CD8s, they're across the board, right? Both in responders and non-responders. So CD8s alone, or measuring CD8s alone, is unlikely to be useful, but it's a good sanity check for us before we go in and do the high-plex spatial analysis. And so Chakra's team handled some of the domain network analysis here. We identified the different microdomains in there. Again, immune cell types, tumor cell markers, structural, functional, metabolic markers. These panels are about 60 to 100 markers, and then the goal is how do we define a minimal signature using these approaches. And so this was interesting. So what we found was that there was this M2 macrophage population that was associated with patients that had recurrence in their tumor, and then when we looked at the patients that didn't have recurrence, we had this anti-oxidative stress linked to anti-inflammatory Treg populations. And so when you look at these on the map, you can see that both different sort of phenotypes are present, and they're clearly defining different microdomains in both responders and non-responders, but also can we develop a minimal signature here? So this is just taking all that data in the head and neck study and defining a multi-marker panel. So we've taken about 70 markers, we've drilled it down into a minimal signature of 6 and 5 biomarkers that define both response and resistance. Granzyme B was one of the markers that came up in this head and neck IO cohort. And what was really fascinating was we see this in non-small cell lung cancer as well. This is a study we've done internally in the lab, independent of PredxBio. And we took all the sort of spatial analysis, and what we found was that the same population, this granzyme B population that's associated with proximity to macrophages, is actually associated with a worse clinical outcome. If you want to see more of the study and the findings from this, please visit the PredxBio booth. And with that, I'd probably hand over to Dusty. Thank you.
Thank you. What an amazing talk. Thank you very much. Well, we're going to hold questions till the end. I'm sure you have lots of questions, but we're going to have the panel right now. So I would invite the panel members to step in and take your seats, and we're going to go ahead and start. All right. We have around close to 30 minutes for the panel. So looking forward to a really interesting and scintillating panel discussion. Okay, maybe let's do a round of introductions. Maybe you start from that side. Well, Arutha, we already talked a little bit about you, but talk a little bit about kind of what you're doing at Queensland that you have not mentioned. Maybe the support you get from your ecosystem, stuff like that. Yeah, is it working? Hey guys, it's not working.
There we go.
Okay.
So I lead a spatial multiomics lab at the University of Queensland. We've established the Queensland Spatial Biology Center, and so that's purpose design. The goal is, can we use spatial biomarkers to really move the needle for translational biomarker studies? And so we're integrating, traditionally we look at retrospective cohorts, but the goal here is, can we identify signatures and then go validate it in proper clinical trials, in prospective trials? There are ongoing studies in the lab, and the lab's probably published 30 to 40 papers in the last couple years on the topic. And so our goal is, how do we drive these signatures to the clinic, define minimal signatures, and then really move it towards CDX type assays?
Hi everyone, I'm Laura Dillon. I'm a Senior Director Partnership Strategy Lead at AstraZeneca. My training's in bioinformatics, but I've been working in biopharma for the last 15, 20 years, specifically in the translational medicine space with a focus on digital pathology, molecular imaging, and radiomics. Right now I'm leading a network of academics at AstraZeneca. We're really focused on identifying how we can use research, it's translational and basic research, to understand and inform strategy for development of our drugs. Using spatial biology is one of the things in our toolkit to do that, particularly multiplex immunofluorescence and spatial transcriptomics, but also other methods.
Chakra, I'm actually a colleague of Tulia up until recently. I was a professor in the Department of Computational Biology. And a lot of this work happened thanks to funding from NIH. And then at some point in time, just before COVID, we spun it off, and then I took a leave of absence, and then be in the company.
Can you hold the mic closer? I think it's you need to hold the mic really close.
Thanks.
It looks like. Can you guys hear at the back what he's saying? No. Okay. So.
Hopefully, I don't have to go again.
Hello? Okay, fantastic. I'll be very loud. Mike Sirachi. I am also at AstraZeneca, and I have a couple of different roles there. One is that I'm the translational medicine lead on a couple of engager and circuit programs where we're responsible for developing strategies for patient selection. The other is kind of the one that's more relevant, which is almost everything multiplex in the area of translational medicine is to one degree or another my area of responsibility. So that's designing, developing panels, validating them analytically, but also looking for solutions for analysis. And I think we're in a very interesting time where we're getting to a point of actually having line of sight on being able to put multiplex into the patient selection space, and that's one of my key focuses for our emerging programs. I am going to be cognizant of the fact that we don't have unlimited time, and I'll pass the microphone to Tulia.
Hi, my name is Tulia Bruno. Hopefully that's loud enough. I'm an assistant professor at University of Pittsburgh and UPMC Hillman Cancer Center. I was pulled into the spatial biology space because my research program focuses on tertiary lymphoid structures, and you can't study TLS in solid tumors without doing imaging in spatial transcriptomics and proteomics. I'd also like to echo that the other reason we really got into this space is because it's very, very clear that where the cells sit in tumors will help to us understand why patients respond to therapies or don't, as you're seeing in some of the data that was presented. So that's a lot of what we do in the lab now, thinking about what happens in treatment-naive patients versus what happens when a tumor microenvironment is treated and reshaped by a specific therapeutic.
Terrific. Have you introduced everyone here? Yes. Okay, fantastic. You know, as I said, I saw around 35 to 40 booths here talking about spatial biology, many of them talking about analytics at this point, but the adoption still remains fairly low across the board. So maybe Tulia, you know, since you work across academics as well as you interact with industry, let's talk a little bit about the adoption and what might accelerate that and what we have learned from genomics, really, for adoption of the technology.
So I think that the spatial biology and transcriptomic, all that space is being really well adopted by academics. I think that what we've been really trying to move past, which I think your group is doing and others, is you've generated this really beautiful picture from a patient tumor. Now what? So how do you analyze that? How do you garner as much information from that so that you can understand what's happening in patients and patient cohorts? I think that, and I think my industry colleagues are in a better position to say where the inertia is or where things are headed, basically for this in the industry side. But I would say from the clinical side, now that we're working in a lot more clinical trials, a lot of this is finding the right markers for the samples that you're getting, right? And I think I see a lot of people shaking their heads. So a lot of what was shown today for me isn't going to work because I'm not going to capture a TLS with a small needle, fine needle aspirate, or a biopsy, right? So how do I get creative in thinking about what I've learned spatially and then applying that to a clinical trial and making that more high throughput, right? And I think that's what a lot of academics think about when they're thinking about clinical trials and also thinking about scaling for these sort of efforts, because it's something that takes a lot of time and energy and money. So I think those are the two places that kind of have been the I guess, restricting it being further integrated into clinical trials. But what I will say is that I'm seeing it more and more. And so I actually push for people to do this because they can usually get more FFP tissue than they can fresh, for example.
Thank you. So, Mike, since you're from AstraZeneca, give us the perspective from the industry about the adoption. How do you accelerate that? And I think you've been a champion of this, I think, for the last 5 years at least. And so you can talk a little bit about the bottlenecks that maybe you're facing, whatever you can share.
Yeah, I think what I'd like to say is that the key to adoption, especially on the industry side, is to understand what incentivizes projects to use these types of technologies. So in the context of the emerging synthetic immunity and expanded sort of second generation immuno-oncology assets, I think it's always a mistake to lead with the tool first, right? So there was a bit of a rush maybe 5 years ago, and we started running multiplex immunofluorescence on everything across a wide variety of trials. And there's a lot of utility to having consistent data across trials, especially when you're talking about a family of assets. However, I think in a lot of cases we found that the bottleneck was, of course, analysis, which I'm very pleased that there are emerging solutions that are available outside of AstraZeneca to smaller companies like biotechs now. But I think the key is to make sure that you have line of sight to impact on the study, especially for actionable insights to make decisions on whether or not the study proceeds, or whether or not you think you understand the MOA, or whether or not you have a better view on pharmacodynamics. So I think that adoption is not what I would the rate of adoption is not what I would consider to be the rate limiter. It's the rate of being able to turn good data, right? Often you have good data into something that matriculates into impact on the study. And I realize I've said that 3 times. Maybe that's the right number.
Thank you, Mike. Maybe Laura, since you've been both in large companies as well as in smaller companies with Incendia, do you see any differences in terms of the technology moving in smaller pharma versus large pharma that you've experienced in the past? And in general, what's your take about adoption?
I mean, there's certainly a lot more stakeholders to align in large pharma than in small biotech. You can move very fast in small biotech, which is quite nice, but being in a larger environment, you have access to potentially to more resources. Just to build on what Mike said, I think the other real big challenge, as we're getting that line of sight for, I'm going to do this experiment to get this answer, which is going to inform how I do my next trial. That's really the type of thing we need to be able to see. But an important additional component really is the timing. And I think that's where a lot of these technologies are so challenging, is that if you have to get your phase 2 or phase 3 data and be doing the experiment in that setting, the ship has already sailed for being able to get to the market. And so I think that's why what's exciting about some of the approaches now, that you can actually look at smaller numbers of patients that you could realistically acquire in a phase 1, starts to make that a little bit more tractable, so that you can make those decisions moving forward in a timely manner to actually impact your drug development pathway. And so that's I think a thing to keep in mind as we're designing these experiments, is how can we actually make sure that we're hitting the outputs when we need to hit them, aligned with strategy in a way that's line of sight and can be communicated across a multifunctional organization, all the way from the early researchers who are really interested in understanding mechanism of action, but also all the way up through regulatory, commercial, bringing everybody along.
Yeah, and Laura, you've been the first ones to really evangelize this whole idea of starting with the high plex in phase 1, 2, and then getting to a much lower plex.
Very happy to see that on your slide.
Yeah, finally we are doing that, right? So thanks again for exhorting us to do that many years ago. Arutha, you work a lot on clinical trials, which is unusual for an academic, lab. But I think that's been the key to your success, because it's not N of 1 that you're dealing with. You're dealing with hundreds and thousands of patients, and your results really have shown the kind of credibility that rarely you see from academia. Go ahead.
Yeah, I think one of the challenges for Spatial has been a lot of N of 1, N of 2 studies. Look at my pretty pictures, and it's great as a sort of an undergrad study, but the challenge is you're understanding the biology not at scale, and so how do we identify the patterns associated with those clinical endpoints? And part of the challenge for us over the last 2 years has been, how do we access cohorts from around the world to be able to validate these signatures, which is what's been part of the challenge. But I think everyone's realized that you can't have single site studies. You need to really be able to, get a cohort from Yale and Brisbane and India and so on, and be able to validate your signatures. And so we're doing all the sort of discovery work retrospectively, but we're also now plugging into prospective clinical trials. And I think that'll really showcase the power of how we can scale these. One way to do it is using tissue arrays, but there's a trade-off there. But I think we'll make some insights into that.
Thank you. And maybe you're also in a unique position to answer this question. I mean, everyone nowadays claims that they're doing AI, right? I mean, I'm sure that you talk to many groups. There are people in academia doing AI. At least they claim to, in all the big pharma companies, big AI groups, foundation models and whatnot. What I don't see is a lot of talk about integrating the cancer biology and AI together, and that's kind of what we've been doing right from day one. Maybe a few comments on how the AI approaches really differ and what exactly needs to be done.
Yeah, it's a double-edged sword, doesn't it? I think we want to get there really quickly, but there's a lot of ground truth. There's a lot of work that we need to sort of do initially. Part of the work we're doing at the moment is integrating your multiplex data with your H&E. I think that's a good starting point. And then you build your omics sort of cloud, and then you can do your imputation and all of that. I think before we get to the spatial agent sort of today-ness of it, I think there's
that's a new buzzword. Everyone is doing spatial agents all of a sudden.
I think there's a ways to go, and obviously, part of the challenges is working with pathologists, really trying to be able to understand, are these cell types we're seeing, are they real? Working with immunologists to really validate them. I don't think we're there yet, Dusty, maybe in a couple of years, but I think there's a lot more groundwork to do in this space.
Maybe Mike Sirachi, you've been, you've been futuristic in your approach from as long as I've known you in terms of where AI can go and what AI can do. Maybe a few comments around what you see at this point across the board.
Sure. From my perspective, I'm trying to identify what this minimal signature is going to be. We think about that in the context of a single project, and it makes a lot of sense, right? We have a large number of targets. We identify a signature, and then we duplex that by there's various methods of minimizing that down to something that's actually deployable. When I think about sort of reasonable applications of artificial intelligence from an agentized standpoint or a mixture of experts approach, I think that I agree with Arutha that we are not there yet. We have a long way to go. We're in a proliferation stage of technologies. I think that the folks who sort of sit and wait and watch what happens are going to very much be at risk of missing opportunities because the rate of advancement in artificial intelligence, just the capabilities of the tools that we all use every day, of the large language models, is so fast that this isn't something that you can wait, look, and pick up once it's matriculated to some sort of point of maturity. This is something that I think we all have to participate in the development. So the way that we're leveraging this in AZ is broad and deep. I'm sure everyone saw the partnership announcement recently between AZ and Tempus from a data science standpoint. That's one example of a commitment that we're making. I think it's also manifesting in smaller ways, such as our approach to identifying multiomics signatures for patient selection, which is at its very early and nascent stages, but is entirely built on a variety of artificial intelligence tools. So I think the short answer is that we're in a proliferation stage, and there's going to have to be novel methods for validating approaches, and then a pruning stage where we coalesce around a couple of key tools and approaches. But the way forward is currently unclear, but there are many opportunities.
I'll come to Chakra in a second, but Tulia, from your standpoint, what do you see in academia? By the way, Chakra came from Jeff Hinton's lab around 20 plus years ago, and he's the one that's kind of incorporated the explainability and cancer biology into the AI algorithms that we have. But we'll come to that. And Chakra, think about the futuristic. What's next? Tulia, go ahead.
Yeah, I think the AI track of trying to analyze samples and really do it this way is going to be something that happens over a very longer period of time. I agree with Mike. I think we want things to happen like yesterday so that it makes it easier to understand what patients should go on which trials, makes it easier to analyze patient samples and bulk predict what's going to happen. But there's just so many different ways to come at this, and there's so many different algorithms and ways in which people are modeling this. I do stand true, though, that I think that the biology that's present in the patient has to drive the AI. I know there's a lot of new kind of let's hypothesize or predict this cell from not thinking about the biology. I think that's going to be really crucial and instrumental, and I don't think we can just generate everything by AI and then hope that that's going to be the answer. I think there has to be a very clear back and forth between the two. That's the one thing that I've seen from those models that have come out recently.
Chakra, future. What do you see happening? The evolution of AI, multimodal data, foundation models, spatial agents. What's your view?
Maybe two comments. And I think
Louder, Chakra, louder, sorry.
Two comments. One, I think if you're familiar with Bayesian thinking, what Tulia just said was about there's this idea that you already know the biology, you want to know if the data verifies it or not. That's like finding the probability of the data given the biology. But what you really need is you want to compute the probability of the biology given the data, because that's the only way you get to this emergent biology. And this is where all these ideas, all the new ideas come in. You'll say, you know what if I build you a very large search space? So all the foundation models, say the LLMs and everything, which is great. But somebody has to go and verify. So I don't think so in this domain, you could talk about AI if it's not actually coming from an immunologist or a pathologist who actually have feel for the experiments, have a feel for the quality of the data, know the pain of actually generating the data, and then if they can articulate how AI is helping, then I would imagine that's a good day. And I think that hasn't happened yet, wherein somebody can actually assure you that these are the things that could come out, and here's a small set of hypotheses that I would like you to verify, and that they could explain that to an immunologist or somebody who's actually working in the lab. So you cannot build a foundation model if your data is no good. And if you have issue with reproducibility, so that means one data set is very different from the other, where is this foundation model going to go? But in many ways, it's all good ideas. If it's about generating hypotheses, generating lots of them, pruning them, fabulous, bring them on. But I think being divorced from experiments, I think it's a danger.
Thank you, Chakra. Sam Hanash, who was there till a few moments back from MD Anderson, is someone that pioneered liquid biopsy almost 30 years ago. He was the first one to show that you can see stuff in blood way before radiologically a tumor manifests itself. And he's been pounding me to integrate spatial biology and liquid biopsy, and that's why I want to ask Arutha, who has worked on both sides, is there some interesting possibilities of integrating liquid biopsy and spatial biology? And we know that there's some activity out there in the M&A space also kind of mimicking that. So let's talk about that.
That's what curveball I didn't see coming. I mean, liquid's great. I think MRD, ctDNA, EVs potentially. I think CTCs are technically challenging. They're rare to find and so on. Liquid definitely has a use case. The challenge is when you identify the change in the blood, but you don't have radiographic data, it's not actionable. And so it's part of the reason I left that field and moved into the tissue space, because we know what we're looking at. And so I think in the future, ideally, how do we do a foundation or garden, tissue and blood and correlate those? That'll be sort of the ideal world. I don't think we're there yet. There's a long way to go. But also, there are signatures we can identify in the tissue that we can mine for in the blood. It'll be highly diluted, but you can, so look for those markers. And so that's the space that we're interested in. Can you do sort of high-plex proteomic profiling of tissue, identify certain targets? There are assays where you can identify, TLS-like signatures in the blood, which, baffles your mind that you can identify it. But if it's there, can we find it? And I think in developing these studies, we need to start to biobank tissue and blood from matched patients. And I think everyone is sort of doing that. And then there'll be a space in which we'll be able to execute on both sort of domains.
Thank you. And Tulia, you've been one of the pioneers in terms of looking at multimodal integration of different modality data. So PET and spatial biology, you want to talk a little bit about that? But that's brand new.
Yeah, so I do want to just say one thing based off what Arutha said. I think the other effort we're making specifically in the TLS space that could also move into like a systemic in using ctDNA is thinking about how the tumor pushes TLS biology and how the tumor pushes the immune response. So we've got new data showing mutational differences based off of when you have a TLS and you don't have a TLS, and then we can then go back to the tumor and say, does that mutation then track in patients? So I think we're a little ways off, but I think that we can also use the tumor to understand the immune, which is, as an immunologist, I know I'm not supposed to be saying, but it actually is the truth, and I've drank the Kool-Aid a long time ago. So you don't have to worry about convincing me about the importance of thinking about the cancer type as well. As far as PET imaging and things go, one thing that we're thinking a lot of from the forefront moving forward, and as I mentioned, I'm very TLS centric, so that's my neighborhood that I think about a lot. We have been thinking about how do we look at these structures in 3D in patients moving forward, right? And so if as hard as 2D is and processing all those images and getting those large data sets together, which, as mentioned, Ruth has probably done this the best out of most that I've seen. There's still that complication in 3D that you can do even less, right? And so finding a way to look at things in 3D, particularly that are important, such as like high endothelial venules, which help with TLS, but help with trafficking of immune cells in general. I think that will be the new frontier. And there's things we're working on related to PET imaging in that space, first in mice, but then thinking about moving into people moving forward.
Terrific. And I'm going to open up this last question for everyone. There's a lot of avant-garde topics coming up at the moment, like AI virtual cell, right? From the Chan Zuckerberg Institute, there was a paper from Stanford, and with 40 other authors around that. So this is all going to a point, can we really simulate clinical trials? Can you run in silico clinical trials ultimately with the network biology that you're exposing from all of this stuff? Arutha, you want to start, and then we kind of just come all the way through back?
Yeah, it's challenging, Dusty. I think it relies on the training data set you've used to really enable that. A lot of the data sets or the models out there today lack clinical annotations, and so the model's developed, but it's developed on no clinical endpoint in that data set. And so, whilst the model exists, there's no utility to it today. But I think, again, looking at the future, this is a space,
Address for a second the universality that you're seeing of some of these biomarkers in combination with others and interacting with each other across different cancers that you're seeing. We talked this morning about lung and head and neck, and what are you saying very quickly?
Yeah. The signatures we're seeing that are associated with resistance or sensitivity aren't tumor specific. They're almost pan-IO specific. Mm-hmm. And so that's really interesting to us. Some of the trends we see in lung cancer, we see in head and neck, we see in cutaneous skin cancers, and we think that's across the board. So some of those signatures of resistance seem to hold across tumor types, particularly in the therapeutic space. So whether it's chemo or IO and so on. So we're starting to think of these signatures as sort of a tissue, sort of a tumor agnostic biomarker, and we think it defines fundamental processes of resistance linked to metabolism and so on.
We're kind of out of time, so quickly, any comments? Anyone else? Go ahead.
I just want to stand up for foundation models a little bit after Chakra is so mean to them. So I think all of these different approaches have a place, certainly. I think to your point, we need to make sure that we're training the foundation models, or that the data that goes into them is appropriately broad, and you spoke to that as well. But really, like, being able to, again, get that timing thing to link up by needing a smaller number of patients, and that's really where those foundation models can have a really important thing. But as we are thinking about going from that broad range of biomarkers down to the smaller amount, we also have to think about how we can get those. And this is where H&E based biomarkers could certainly play a role, because everybody in their gets an H&E, absolutely. This is where the serum biomarkers or even the PET based ones are so important, because those kind of less invasive, non-invasive type of methodologies are indeed what's better for patients. Mm-hmm. And so it's hard to talk about these in generalities, because like every use case is a different use case and needs a different answer. But I think there's roles for all of this, and certainly the technology we're talking about here today is hugely impactful for helping to understand that kind of broad space, so that we can understand the biology and then go back and really drill down on, okay, how can we come up with an actionable plan that we can deploy in the clinic to bring drugs to patients, not just in clinical trials, but actually in clinical practice.
Thank you, Laura. Mike, 30 seconds, you want to use?
Yeah, I'll be as brief as I can. I think just quickly on the question of an in silico clinical trial, I think it's not only possible but inevitable, but it's contingent on a model that is validatable, sufficient processing power, and sufficient data to train that model. So foundation models are an example of something that can be trained on disparate and deep data that's not necessarily all relevant to the question, but then you can use those to answer questions that were not part of the original data set with relative utility. So the short answer is yes, but the longer version of that is I think it's contingent on substantial advances that I expect, but when those will matriculate.
Thank you, Mike. Thank you. Well, as Yogi Berra says, prediction is always difficult, especially when it's about the future. So on that note, thank you very much. Really appreciate your participation, and a big hand for the panel.